Hello and welcome to Nikolai's Genetics Lessons. My previous 3 videos were about Manhattan plot. This is what you see here. But today we are going to talk about this plot which you see here in the right top corner, which is called QQ plot. So today we are going to talk about evaluating of the quality of the genome-wide association study results. Primarily depends on the QQ plot, which you see here. Before I will explain how to read and analyze QQ plot, let me first tell you in few words what is a Manhattan plot is. On the y-axis, here we see minus log 10 of the p-value, and on the x-axis we see chromosomes. In this case, 22 autosomes. Take a look, here is a human karyotype, and we have 22 autosomes and 1 pair of sex chromosomes. So autosomes goes according to their size from the biggest to the smallest. That's why here the width of the chromosomes, as you see, also going to reduce to the end of this row, because on the chromosome number 1 there are more genes than on the chromosome, for example, 22. As you see here, chromosome 22 is much narrower than chromosome number 1 or 2. Now take a look at this structure of the chromosome, and you see here centromere. That explains this gap here, which stands for absence of genes here, and hence single nucleotide polymorphisms, which we are looking for. You also can see gaps between chromosomes because caps of each chromosome have highly repetitive DNA, which doesn't also code for any proteins. So in these regions we also don't have single nucleotide polymorphisms. We are mostly interested in single nucleotide polymorphisms within genes. They can be in introns, exons, or close to genes, or close to regulatory regions. So as you see, we are using genome-wide association study as a powerful research method used in genetics to identify associations between specific genetic variations and particular traits such as disease, physical characteristics, or it also can be, for example, association of single nucleotide polymorphisms with athletic performance. So researchers collect DNA from thousands of people, or it can be plants or animals with specific trait, and compare it to DNA from similar group without that trait, which we call controls. Instead of looking for a single gene, genome-wide association studies scan entire genome for small variations called SNPs, or single nucleotide polymorphisms, and results are reported in a form of Manhattan plot, which got a name because it resembles skyline of the Manhattan. A computer checks if any specific SNPs is found more frequently in people with a trait than in those without it. Genome-wide association studies has revolutionized medicine and biology by allowing scientists to identify genes involved in a complex diseases like diabetes, Alzheimer's, or heart disease. It also allows to understand the genetic basis of athletic traits such as muscle power or endurance, and develop personalized treatments based on person's unique genetic code. Now let's return to our QQ plot. On the x-axis here we have expected minus log 10 of p-value, and on the y-axis here we have observed minus log 10 of the p-value. So we're comparing your data with average population. So what this red line represents, it represents a null hypothesis. It shows what the data would look like if there were zero genetic associations. Essentially, if everything was just a random chance. If all blue dots stayed on this red line, that means that your study found nothing. What we have to pay attention here is a tail, which here shown in the red box. Notice how the dots at the far right on this image curve sharply upward and away from the red line. This is exactly what you want to see. This tail represents the true genetic signals, the skyscrapers on this Manhattan plot. It means that these SNPs are much more significant than what would happen by random chance only. It's also important to understand that if blue dots lift off the red line right from the start, near the bottom left, it usually means population stratification. In plain English, that means that results are biased because of the ethnic makeup of your groups or cryptic relatedness. For example, you accidentally included a bunch of cousins rather than actual biology. But what if the dots stay below the red line? That means that your model is too strict. You are killing your signals and you might miss real discoveries or false negatives. So the perfect plot would look exactly like on this picture. Most dots stay on the line and only most significant ones curve up at the end. This suggests a clean study with real hits. Now let's take a look at more examples of QQ plot that you might get, and the one which we already analyzed is here. Take a look yourself. The first and second plots deviate early and have high false positives, which is not good. The third plot looks pretty good. The fourth plot is somewhat off, and fifth plot is great. The sixth plot lifts too sharply at the end, possibly too many false positives. And the seventh plot is also good. Hence, we can come to conclusion that plot number 3, 5, and 7 are relatively good, with plot 5 being nearly perfect. And this is all for today. Subscribe and see you in the next video. Goodbye.